Welcome back, everyone. Next up, we have Humacyte Inc. It trades on the NASDAQ under the symbol HUMA and is developing a disruptive biotechnology platform to deliver universally implantable bioengineered human tissues, advanced tissue constructs, and organ systems designed to improve the lives of patients and transform the practice of medicine. The company develops and manufactures acellular tissues to treat a wide range of diseases, injuries, and chronic conditions. Humacyte's initial opportunity, a portfolio of human acellular vessels, is currently in late-stage clinical trials targeting multiple vascular applications, including vascular trauma repair, AV access for dialysis, and peripheral arterial disease. Please welcome its founder and CEO, Dr. Laura Nicholson, and CFO, Dale Sander. Welcome. How are you all today?
Thank you very much. We're great, and we really appreciate the opportunity to participate in this forum. I'm very excited to be here.
Wonderful. Well, the floor is yours. We are excited to hear your presentation.
All right. So after that introduction, I'm going to provide essentially these are the standard disclaimers, given that we are a publicly traded company, and these forward-looking statements should not be viewed as promises of future outcomes. As you heard in our introduction, Humacyte is really a first-in-class regenerative medicine company that is working to develop engineered human tissues that are really absolutely unique. Humacyte is the only company on the planet that's making tissues that have these uses in this way. Our engineered tissues are available off the shelf. Importantly, it's not necessary for us to take cells from the patient in order to make a tissue for a patient. Humacyte has a working cell bank that we use to manufacture our human tissue replacements at commercial scale. Because we are a universally implantable tissue, what this means is that we're able to implant our tissues into any human recipient without rejection. Also importantly, once our tissues are implanted, they repopulate with cells from the patient and become a living tissue over time. In fact, these tissues become self. A few highlights about Humacyte include the fact that we have large total addressable markets because our platform technology truly is a platform, and we can make tissues in different shapes and sizes with different mechanical characteristics. We're able to make tissues that address a large number of diseases, and so our worldwide TAM is estimated at around 150 billion. For our first indication, which is using our engineered vessels to treat acute traumatic injuries, we expect to file a biologics licensing application, or BLA, with the FDA sometime in the middle of 2023, and therefore have approval and be on the market in the first half of 2024. Humacyte was the first company to receive the FDA's Regenerative Medicine Advanced Therapy, or RMAT designation. We're also a priority product by the Defense Department, and you'll hear about why this is in a few minutes. Importantly, we have a commercial scale modular manufacturing that's already in place. In fact, for our phase 3 clinical trials, we're using our ultimate commercial scale system to provide clinical product for our ongoing studies. And as mentioned, we trade as HUMA on the NASDAQ. This is our leadership team, which is very experienced and in many cases has been with the company for a long time. We also have a superb board of directors, chaired importantly by Kathleen Sebelius, who was the former Secretary of Health and Human Services in the Obama administration. So here's a summary, just a high-level overview of our manufacturing platform process. As I mentioned, we start with a bank of human cells that Humacyte has developed over a period of more than a decade. This cell bank provides essentially the building blocks or the raw materials that we use to make our engineered tissues for all of our applications. When we want to make a batch of human blood vessels, we thaw a vial of cells and expand them, and then seed them onto a biodegradable polymer scaffold. Now, the size and the shape of the scaffold really dictates the size and the shape of the tissues that we ultimately grow. And that's why this is a modular and scalable platform. After the cells are seeded, they grow on the scaffold for a period of about 2 months. And during that time, the cells secrete extracellular matrix proteins like collagen. And while that's going on, the scaffold underneath them dissolves. So after 2 months, what we have is an engineered human tissue that we've grown from scratch that consists of cells and the matrix proteins that they synthesized. However, the polymer scaffold is now gone. In a final step, we wash the cells out of the tissue. And what that does is it removes the immunogenicity of the tissue, making it implantable into any human recipient, and also gives the tissue a shelf life because it's non-living. Currently, our shelf life is 18 months. This is just a summary of some of the markets that we're addressing in both our clinical and our preclinical studies. We currently have clinical trials going on using our 40 centimeter by 6 millimeter HAV to treat patients with kidney failure who are on dialysis, to treat patients with peripheral arterial disease, and also patients with vascular trauma. In addition, we have preclinical and primate-based work underway using different configurations of our vessels to treat heart bypass for coronary artery bypass grafting, also a model of pediatric heart surgery, and also a primate model of the treatment of diabetes, which I'll talk about a little bit later in this presentation. So we have late-stage phase 3 clinical trials underway in 2 indications, in vascular trauma and in dialysis access. And as I mentioned, our application in vascular trauma is the first one that we plan to advance with the FDA. In addition, we have a phase 2 trials underway and in long-term follow-up in peripheral arterial disease. So to give you a little bit more sense of what the human acellular vessel, or HAV, is like, you can see an image of it here. It looks like a white plastic tube if you were to handle it. It feels like a native human artery. For those of you who know what those feel like, if you don't, then it feels a little bit like calamari. It's a rubbery, mechanically very strong tissue that is nonetheless acellular and non-living. It implants into patients very similarly the way vascular surgeons implant other types of veins or arterial grafts into their patients. Importantly, after the HAV is implanted, it repopulates with cells from the patient over time. This is just an example of that phenomenon. This is an explant from a patient who was a dialysis patient who got the HAV for hemodialysis. This is a biopsy that we took about 9 months after implantation. You can see in the middle of the image that there are many red spindle-shaped cells. These are all cells that stain for a smooth muscle protein called alpha-actin. This is a protein that's expressed by cells that are in the walls of your arteries. You can also see a lot of small blood vessels or capillaries surrounding the outside of the HAV. But what's important to know here is that when the HAV was implanted into this patient, if you had looked for any smooth muscle markers in that HAV, they would not have been there. So all of the cells that are in the HAV migrated into the vessel from the patient's own stem cells. So again, in summary, the human acellular vessel, or HAV, is available off the shelf because it's decellularized and has a shelf life of 18 months. We've shown in our clinical experience, because we've been in man for more than 10 years, that the HAV is highly durable in multiple clinical indications, including peripheral arterial disease, dialysis, and trauma. We've seen no evidence of immunogenicity in over 500 patients implanted. And we've also seen across our clinical studies that the HAV seems to be highly resistant to becoming infected, which is particularly important if you're treating patients with traumatic injuries, because many of those wounds are contaminated. So this brings us to our next section, which is discussion of where we are in vascular trauma and why the Defense Department was so keen on making us a top 5 priority several years ago. So sorry for the gory photos, but this is the image on your left is an image of a soldier who was fighting in Afghanistan who was injured by a blast, by an IED explosion. This soldier obviously had damage to his thigh and to the artery that supplies the leg. Under normal conditions, what a surgeon would do to repair that artery would be to take vein out of the other leg and spend an hour or so taking that vein and then move it over and repair the injured artery. Unfortunately, this particular soldier lost the other leg in the blast and so had no place from which to take vein. So in that case, surgeons were forced to take a plastic graft made out of PTFE or Teflon off the shelf and use that as a repair for that injured soldier's artery. Not surprisingly, when you put a piece of plastic into a contaminated wound bed, the plastic graft became infected, and that soldier wound up losing his other leg. So for these reasons, the Defense Department places a very high priority on having engineered tissues, such as blood vessels, that they can pull off the shelf, use immediately without any delay, but know that when they are implanted into contaminated wound beds, that they will resist infection. And so because of that, the Defense Department has helped support our ongoing phase 3 trial in this space. But beyond even the military injuries, the civilian vascular trauma market is actually substantially larger. And the limitations in treating patients with civilian trauma, for example, from car accidents or gunshot wounds, are very similar to the military case. So when surgeons need to revascularize an injured limb, they need to do so quickly, and they need something that's going to resist infection, because the consequences of delayed revascularization, which can lead to amputation or infection, are both bad for the patient and costly for the system. So as I mentioned earlier, we have an ongoing phase 3 trial that's in the later stages of recruitment. Importantly, this is a single-arm trial, so this is not a prospective randomized study, but rather this is a trial in civilian trauma patients who present to the emergency room needing vascular repair. And these are all patients for whom autologous vein is really not an option because they don't have it or because there's no time to harvest the vein. In these cases, these patients are consented and receive the HAV to treat their wounds. What we've seen in this non-randomized open-label study is that the HAV shows very high patency rates and good rates of blood flow. We've had very low rates of infection. In fact, out of more than 60 patients, we've seen only one infection, which is remarkably low given the 20, 30, or 40% infection rate that you might expect with synthetics. We've seen low rates of amputation and no amputations that were caused by a failure of the implant. And we've also had no rejections. Going further, looking at the function of the HAV in trauma in a real-world setting, it's important to know that starting in March of last year, Humacyte began receiving requests from surgeons in Ukraine to provide HAVs as treatment for wounded warfighters in that conflict. Humacyte worked with the FDA and with the Ukrainian government to ship HAVs to 5 frontline hospitals in Ukraine. And since June of last year, we've treated a total of 13 patients, many of whom have contaminated war-caused blast injuries that have caused arterial damage and caused risk of loss of limb and loss of life. I'm very glad to say that out of the 13 patients we've treated, we've seen 100% success with no infections and no losses of limb or patency. Again, sorry for the gory photos, but this is actually the first patient we treated in Ukraine. This is a patient who suffered a gunshot wound and then got a plastic graft to repair that injured artery, which you can see on the image on the left, that sort of corrugated tube in the middle of the image. That tube became infected and the soldier was at risk of losing his limb, and so he was transferred to a hospital that had the HAV. The HAV was implanted in the leg, and we have video of that soldier walking out of the hospital several months later. So this is just a real world example of how the HAV functions very well in these real world conflict conditions. In the interest of time, I'm going to talk a little bit about some of our pipeline activities because I think they may be of interest to this audience. I'll begin very briefly with a summary of where we are in developing a smaller caliber version of our graft for coronary artery bypass or CABG surgery. So as many of you may know, it's very common to take veins out of the leg to do a coronary artery bypass operation. But that procedure in itself can cause a lot of complications, and there are many patients who don't have suitable vein for this operation. So Humacyte has developed a smaller caliber version, a 3.5 millimeter version of the vessel that we're currently studying in a primate model of heart bypass. What we've seen using our human acellular vessels in a primate heart bypass model is that the vessels are very durable, and we've shown that they have good function out to 6 months. We've also shown that just like vessels that I showed you from our clinical patients, vessels that we implant in the heart in baboons also repopulate with cells and become living arteries over time. These results have been reported at the American Heart, and our ongoing large animal studies that we're continuing in 2023, we hope will contribute to an IND filing to go into phase 1 in coronary artery bypass in later years. In addition, we're using a modification of Humacyte's acellular vessel as essentially a delivery vehicle to bring islets to patients who are suffering from type 1 diabetes. As many of you may know, the islets, which are the insulin-producing cells in your pancreas, are killed off in patients with type 1 diabetes from an autoimmune reaction. In order to replace those insulin-producing islets, for 20 or 30 years, surgeons and physicians have been trying to take islets from pancreases and inject them into patients in different locations in the body to try to re-provide that capacity to produce insulin. The problem is that when you inject islets into the liver or underneath the skin, in most cases, many of those islets die because they don't have enough oxygen and nutrients to support them. So the Humacyte concept of the biovascular pancreas is to use our current vessel, which we've implanted in 500 patients, and to coat the outside of that vessel with pancreatic islets that may even be derived from stem cells, and then to implant that coated vessel into the arm of a patient between an artery and a vein. This operation is exactly analogous to what we've done in dialysis patients for many years. The substance of the idea has been supported by work that we've done and published in primates, showing that if we make a small version of the biovascular pancreas and implant that into the aortas of primates who have type 1 diabetes, that we can reverse their diabetes and normalize their blood sugar over time. That's shown by the blue curve on the right, which contrasts to the red curve on the right, which shows animals that got the BVP but got it just implanted into the abdomen and not in the circulation. This really shows the value and the importance of bringing a therapeutic number of islets right next to the circulation, which is what we believe the BVP will be able to do for patients. So as a follow-on to this, we've begun primate studies of this concept, and we anticipate moving forward with that during 2023 and having continued news flow there. And so with that, I'm going to turn it over to Dale. Hopefully I haven't taken up too much time, and have him talk about commercialization and some of our path to market.
Hey, I'll control the slides.
Oh, okay. Sorry.
Yes. Yeah. Thanks, Laura. As Laura mentioned, we expect to file for approval this year in vascular trauma, which would put us in the market in 2024. Within the United States, our plan is to market the HAV directly ourselves. Our launch in vascular trauma targets a very concentrated market. There's only about 200 level 1 trauma centers within the U.S., and it can be reached with a sales force that's relatively small, about 20 representatives. Beyond that, we'd also expect the Department of Defense due to the high need within warfighters that Laura mentioned to stockpile the HAV for deployment to future sites of conflict. Beyond that, we have a major collaboration with Fresenius Medical Care. FMC, as they're known, is the largest provider of dialysis services within the US and other major markets around the world. They're not only a collaborator, they're also a major shareholder of ours. They've invested $175 million in Humacyte. They're our second largest shareholder. And really their interest from an investment point of view is the potential impact on dialysis in terms of cost savings and improvement of patient care. We had to skip over the dialysis section today due to length of time of this presentation, but that is our second indication of anticipated launch with the HAV. And Fresenius serves as a great partner for us within the United States. They've committed to adopt the HAV as standard of care in those dialysis patients that are targeted by our clinical studies. And outside the United States, particularly in Europe, they'll serve as our marketing partner for our first 3 indications, which are the HAV used in vascular trauma, AV access for dialysis, and peripheral artery disease. So within our manufacturing space, we've heavily de-risked this. We're currently producing the HAV for use in our clinical studies and our humanitarian efforts within the same facilities that we'll use for the commercial launch of the HAV. In 2021, the FDA allowed us to progress from our earlier stage manufacturing techniques and facilities into the commercial scale facility that you see shown in these slides. And so we are already producing at the same scale that we use for commercial launch. Laura mentioned the production process for the HAV, the seeding of the cells on a scaffold, and the production, ultimately the decellularization that allows this to be implanted in any patient. The actual production of each HAV is done in what's called a bioreactor bag, which you see in the upper left-hand side. And through the entire production process, through shipping and storage at the hospital until it's opened in the surgical suite, it remains in this bag. These bags are arranged into a set of 200, which is produced in our Luna 200 machine. We have 8 of these machines in place already within our facility, which we believe will handle the market requirements for the first several years after launch. So 2023 is going to be a very busy year for us. Looking at vascular trauma itself, we expect to complete our phase 3 trial, release the top-line results, file our BLA with the FDA, and have that BLA accepted by the FDA, leading to an approval and commercial launch in 2024. As Laura touched on, this is a very broad platform. We have a number of other programs ranging from preclinical all the way up to phase 3, and we'll have a number of data releases, scientific meeting presentations, and publications around the remainder of the pipeline also. So with that, I think we have a few minutes. We'd be happy to respond to any questions that anyone has on Humacyte.
Thank you so much. Yes, we do have some questions. So talk a little bit about how surgeons compare the feel and handling of your HAV compared to the patient's own blood vessels.
Well, I would say that surgeons, almost to a 1, say that they are surprised at how well the HAV handles. Most surgeons will tell you that it's a little bit easier to handle than vein, and actually handles very similarly to a young, healthy, native femoral artery, for example. So most surgeons find this much easier to handle than a synthetic graft or even the native vein.
And so after your approval for the treatment of vascular trauma, are there circumstances in which you think that vascular surgeons will use the HAV on an off-label use?
Well, Humacyte would, of course, never advocate for off-label use, but I can say that the realities are that if you have a vascular conduit on the shelf in the operating room, surgeons will tend to reach for it if they think it will benefit their patient. So if there are patients who have contaminated wounds that are not related to acute trauma, or patients who need some other revascularization, it's possible and it's even likely that surgeons will reach up and use the product where they think it can benefit their patients.
And do you have adequate manufacturing capacity to meet the expected demand after this FDA approval and market launch?
Yeah, with the equipment we have in place, we believe we can produce approximately 8,000 HAVs per year. That represents about $200 million plus worth of production. So we would expect that, particularly for the initial launch in vascular trauma, we can meet the needs of several years worth of market demand. Beyond that, the building itself has capacity to expand the number of Luna 200 machines we have up to 40 machines, which would allow production of 40,000 HAVs per year. So within the facility itself, we can certainly meet several years and beyond that worth of production requirements.
And who are your largest shareholders?
It's certainly a mix. We're certainly quite pleased with the shareholder group we've had. They've been very supportive both during the private and public stages. Our largest shareholder is Brady Dugan, the former CEO of Credit Suisse Bank. Our second largest shareholder is Fresenius, who's our collaborator. And our third largest shareholder is a large family office that is based over in Europe.
And we have a question from John Zhang. He wants you to discuss the schedule for FDA approval for your HAV products. Just go back over that, please.
Well, we've been working closely with the Center for Biologics and the OTAT office there regarding our trauma indication. We anticipate, and we've messaged the market, that we will submit our BLA for the trauma indication sometime in the middle of this year. The exact date is not yet finalized, but we anticipate middle of the year. If the FDA complies with their PDUFA guidelines as far as time of review, then if we submit in the middle of this year, we would anticipate approval sometime in early 2024.
And Richard Martin asks, is nanotechnology a part of your growth strategy? And if so, what future investments are you proceeding with? In other words, is it cancer, heart disease, mental disease?
So yes, so certainly you can argue that our tissue engineering technology is inherently a nanotechnology because the matrix molecules that we make are all at the nanoscale. They're all engineered by cells, not by us, because frankly, the cells are smarter than we are. But everything that we make has a nanotechnology component to it. We're certainly looking at branching out into a variety of other diseases and other organ systems, including the respiratory system, elsewhere in the cardiovascular system, potentially the GI tract and also the urinary tract. I will say that the brain is probably one place where we're not contemplating going yet, although we are contemplating in the long term doing whole organ regeneration. In fact, we count engineered lung or engineered lung lobes as one of our long-term pipeline opportunities.
Thank you for that answer. We have a question from Dr. William Walkstein asking about the biologics division of the FDA. Has it signed off on a definitive number of patients needed to accrue the lower extremity trauma trial, which would be acceptable to submit a BLA?
We're continuing to have discussions with the FDA on the final patient number. As we have messaged to the market for about the last year and a half, we have been anticipating that roughly 75 total treated patients would be sufficient to file a BLA. I can tell you that in our VO5 trial, we've enrolled more than 60. And in Ukraine, which was not something that we had contemplated even a year and a half ago, we've treated now 13 patients. So we're now approaching a total of 75 patients. And we continue to believe that this number is going to be what is required in order to file a BLA sometime in the middle of this year.
Wonderful. Well, we might get one more question in before time is up. Ken Kamikaze asks, any big milestone payments expected anytime soon?
Yeah, I'm not sure if those are incoming or outcoming payments, but no, we don't expect milestone payments either way occurring in 2023. So certainly we get plenty of grant support. We have the ability to partner some of our earlier stage programs, but nothing is set in stone at this point in time.
All right. And Daniel Redant asks, is the cost competitive? Last question.
Certainly cost when you consider the total cost of treating the patients. I think using trauma as an example, regardless of where we end up announcing a price on this, our discussions with hospital administrators would suggest our budgetary impact, which means avoiding the cost of amputations, infections, length of stay in the hospital, will be a positive outcome for the hospital.
Well,
positive net outcome, I should say.
Well, thank you, Dale and Dr. Laura. Really fascinating stuff you have going on here. We would love to have you back in the future with some updates. Thank you for your time and presentation and hard work on this.
Thanks.
Looking forward to it.
Okay, thank you. Okay, everyone stay with us. We'll be right back with our next presenter.